Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing GAT B entrance exam question paper conducted in the year 20. Coming to the first question, the most abundant enzyme present on earth. So it is rubisco. It is present in cyanobacteria to the leaves of huge trees. So the answer is option 1, rubisco. Next one, which of the following statement about enzyme is correct? They are biological catalysts. That is correct. Lactose is made of glucose and galactose. The causative agent of sleeping sickness in human beings is, so the extracellular parasite Trypanosoma brucei causes human African trypanosomiasis, also known as sleeping sickness. So it is an extracellular parasite present in blood plasma. Option 3. Then next one. The cell organelle abundant in liver for detoxification of drugs and toxicants is, so it is the smooth endoplasmic reticulum. Then proinsulin matures into active insulin by removal of the C-peptide sequence. The enzyme used to seal the sticky ends of restriction fragment is DNA ligase. The enzyme encoded by LAC-Z gene in the LAC operon helps in. So the gene product of LAC-Z is beta-galactosidase, which cleaves lactose, a disaccharide, into glucose and galactose. So break down lactose into its monomers. Answer is option 1. An elaioplast is involved in. So elaioplast is a leucoplast that is primarily involved in storing fat or lipids inside fat droplets in plants, especially in monocots and liverworts. So it is storage of fats or lipids. Answer is genome of HIV is, it has 2 copies of positive and single-stranded RNA genome. Answer is option 4, single-stranded RNA. Choose the correctly matched pair of causative agents and the diseases they cause. So bacteria, it causes AIDS. Bacteria doesn't cause AIDS. AIDS is caused by a virus. Syphilis is caused by bacteria, and typhoid is also caused by bacteria. Measles, mumps and polio, they are caused by virus. So that is correct. Elephantiasis is caused by helminth. Ringworm is caused by fungus, and yellow fever is by virus. So that is also not correct. Protozoa. Chickenpox is caused by virus. Sleeping sickness is caused by protozoa, and cholera is caused by bacteria. So the option that is correct is by option 2. The S in the ribosomal subunit represents. It represents Swedberg's unit or sedimentation coefficient. It is differential sedimentation rate during centrifugation. Then next one. Excessive fasting by a human being will lead to the presence of the ketone bodies. Answer is option 2. Then RT-PCR is used in the study of RNA expression levels. Option 3. Then in a dihybrid cross, Mendel's law of independent assortment can be best described by. So it is option 4. Capital Y, capital R, small r crossed with capital Y, capital R, small r. The common structures in a bacterium, plant and animal cell are. So they are plasma membrane, ribosomes and cytoplasm. Option 2. The molarity of a solution prepared by dissolving 8 gram of NaOH in 250 ml of water will be. So molarity is given by formula 1000 into Wb by Mb into V, where V is in ml. Substitute the values and you will get 0.8 molar. Answer is option. How much NaCl is required to make 10 ml of 2 molar NaCl solution? So use the same formula. Then you have to calculate for Wb. How much it is. Substitute in the formula and you will get 1.17 grams. Which of the following is an antibiotic? So it is chloramphenicol. Aspirin is an analgesic antipyretic, where Valium is for sleeping pills, and chloramphenicol is the antibiotic. And Ciplantin, it is an anti-tumor agent. Then in ELISA technique, the formation of product is monitored as change in color intensity. Protein transport into endoplasmic reticulum cell is powered by GTP hydrolysis. Then next one. What is the number of hydrogen bonds in a double helical bDNA structure of 100 base pairs with 20 adenines and 10 thymines in one of the 2 strands? So there are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. If there are 30 A plus T in one strand, there must be 70 G plus C residues in the same strand of DNA. So total number of hydrogen bonds will be 30 into 2 plus 70 into 3, that is 270. So answer is option 1. Then percentage of sequence identity for the following aligned sequences. Now percent identity is the number that describes how similar the query sequence is to the target sequence. Or I can say how many characters in each sequence are identical. So here 7 characters are identical. 1, 2, 3, 4, 5, 6 and 7. 7 out of 10 are identical. So it is 70% identity. Then next one. Which organelle is not present in an isolated cell protoplast? So protoplasm of a living plant or bacterial cell whose cell wall has been removed. So cell wall is not present. Then next one. Which of the following is the most abundant leukocyte? It is neutrophil. It makes about 60 to 70% of the total amount of white blood cells. Then next one. Carbohydrates need 7.6 billion world's human population are provided by the members of a single plant family. Which plant family it is? It is Poaceae, where you get rice, wheat, etc. It belongs to Poaceae. Then fatty acids cannot be converted into carbohydrates in the body as the following reaction is not possible. So fatty acids and ketogenic amino acids cannot be used to synthesize glucose. This is because the acetyl-CoA produced as a result of beta oxidation cannot be converted back to pyruvate. So the answer is option 1, conversion of acetyl-CoA to pyruvate. Then next one. Which of the following statement does not hold good for microbial production of penicillin by Penicillium notatum? The process follows growth-associated product formation kinetics. Then next one. Select the set of fat-soluble vitamins. So A, D, E and K are fat-soluble vitamins. So option 1 is the correct answer. Then next one. mRNA in bacteria bind to ribosome through the Shine-Dalgarno sequence. It is a sequence of 5 to 9 or approximately 7 nucleotide located upstream of the start codon in prokaryotic mRNA that is recognized by the ribosome. Answer is option 3. Then next one. Match the products listed below with the enzymes used for their commercial production. So aspartame, thermolysin, lactose-free milk, beta-galactosidase, cheese, chymosin, cocoa butter substitute, lipase. Answer is option 3. DNA polymerase 1 has all the following activities except. So DNA polymerase 1 possesses 3' 5' exonuclease activity or proofreading function which lowers the error rate during DNA replication and also contain 5' 3' exonuclease activity which enable the enzyme to replace nucleotides in the growing strand of DNA by nick translation. There is also 5' 3' DNA dependent DNA polymerase activity. So 5' 3' exonuclease activity is there, 5' to 3' polymerase activity is there, 3' to 5' exonuclease activity is also there. So all the given options are correct. Then next one. Plant gum is composed of different polysaccharides except starch. Then nitrogen fixation is the conversion of nitrogen to ammonia. Then malaria disease is caused by protozoa. It is caused by Plasmodium parasites which spread to people through the bites of infected female Anopheles mosquitoes. And Plasmodium, it is a protozoan. Then next one. During one round of TCA cycle, how many molecules of carbon dioxide are released? So the first decarboxylation produces alpha-ketoglutarate from oxalosuccinate in a reaction catalyzed by isocitrate dehydrogenase. The second produces succinyl-CoA from alpha-ketoglutarate in a reaction catalyzed by alpha-ketoglutarate dehydrogenase. So total 2 molecules of carbon dioxide are released in one turn of citric acid cycle. So answer is 2. Then which of the following is a sequence alignment tool? It is BLAST. Basic Local Alignment Search Tool. BLAST. Then what is BR in PBR 322 plasmid? It is Bolivar and Rodriguez the two scientists who constructed the PBr322 cloning vector in 1970. Then which of the following molecules is not a secondary metabolite? It is malonic acid. If the genotype of an individual is as given here, how many types of gametes will be produced by this individual? So this is the genotype. So number of heterozygotes in the genotype, that is this one, and B and C are only heterozygotes. So only 2 are there. So number of gametes is given by the formula 2 raised to n, where n is the number of heterozygotes in the genotype. So 2 raised to 2, that is 4. Answer is 4. Which among the following is a character-based method for construction of phylogenetic tree? So two character-based methods are maximum likelihood and maximum parsimony method. So maximum parsimony is given in the option. So that is the answer. Phosphofructokinase is allosterically regulated by several effectors. Which of the following would allosterically inhibit the enzyme? So a phosphofructokinase is allosterically inhibited by high levels of ATP. So answer is option 1. Why is mitochondrial DNA considered to be the best marker for phylogenetic analysis? Because mitochondrial DNA shows maternal inheritance. The next one, the genome size of M13 bacteriophages. So the genome of this phage consists of 6.4 kb single-stranded positive sense circular DNA and encodes 10 genes. So the answer is 6.4 kb. Which of the following amino acid is most hydrophobic? So it is isoleucine. Serine is polar, cysteine is hydrophobic, asparagine is polar. But they have asked most hydrophobic, so it is isoleucine. Isoleucine is one of the most hydrophobic amino acid because the R group is a bulky substituent lacking any hydrophilic character. The next one. The primary RNA transcript of the chicken ovalbumin gene is 7,700 nucleotides long. But the mature mRNA that is translated on the ribosome is 1,872 nucleotides long. This observation suggests that this gene is. So a split gene is the answer. So a split or interrupted gene is defined as a gene consisting of introns and exons. Removal of the introns from a primary transcript is essential for creating mRNA. It is because of the removal of introns from primary RNA transcript that the size of mature mRNA decreased. So the answer is option 1, that is split genes. Then, which of the following incorrectly describes a molecule with a mass 1000 times that of water? So molecular 18,000 daltons, molecular weight 18,000 daltons. If MR is written, then you need not have to write daltons. So the answer is option 2. A TCA cycle operates under aerobic condition. Reaction catalyzed by which of the following enzyme requires participation of molecular oxygen? So none of the TCA cycle enzyme require participation of molecular oxygen is the correct answer. Several RNA viruses form replication complexes by utilizing membranes from endoplasmic reticulum. Then, in a vector, HIN3 and BAMH1 restriction sites were present in the ampicillin resistance gene, while EcoR1 and XHO1 sites were present in the chloramphenicol resistance gene. A student cloned his insert DNA fragment using HIN3 and BAMH1 sites. How will he select his recombinants? So, by plating transformants on a plate containing both the antibiotics, even the non-recombinants will grow. So that is not the best option. Recombinants cannot be differentiated from non-recombinants. They can be differentiated. Plating the transformants on a plate containing ampicillin and then replica plating on chloramphenicol containing plates. So when plate them on plate containing ampicillin, no colonies of recombinants will be formed as the gene of interest or the insert DNA is present in the ampicillin resistance gene region. So it will not form recombinant colonies. Now, next option. Plating the transformants on a plate containing chloramphenicol and then replica plating on ampicillin containing plates. So when you plate them on chloramphenicol, both recombinants and non-recombinants will grow. Now when you replica plate them on ampicillin containing medium, the recombinants will not grow. To know which is the recombinant, match the ampicillin containing plate with chloramphenicol containing plate. Now this is the ampicillin containing plate, this is chloramphenicol containing plate. You replica plated from chloramphenicol into ampicillin. Now in ampicillin plate you can see that 3 and 7 has not grown. So that means 3 and 7 are recombinants. Then the bond length of a peptide bond is 1.32 Armstrong. Addition of salt to a culture medium allows only salt-tolerant bacteria to grow. This is an example of selective media. Since addition of salt to a culture medium allows only the salt-tolerant bacteria to grow, this is an example of selective media. Then, a coenzyme or a metal ion that is tightly or even covalently bound to the enzyme protein is called prosthetic group. Which of the following amino acids give yellow color on reaction with ninhydrine? Except proline, all the amino acids are oxidized by ninhydrine to give purple color. Proline gives yellow color because it is secondary amine. Answer is option 4, proline. Sequence comparisons do not provide information about. So sequence comparison, they do not provide information about gene locations. Which of the following interaction are involved in stabilizing the structure of proteins? So ionic bonds are there, hydrogen bonds and hydrophobic interactions are there. So all of the given options are correct. Then Louis Pasteur is not associated with discovery of Mycobacterium tuberculosis. It was discovered by Robert Koch. Next one. Which of the following statement is true for the reversible competitive inhibition of an enzyme catalyzed reaction? So the value of Michaelis-Menten constant increases. Now why does Km increases? The reason is that the competitive inhibitor is reducing the amount of active enzyme at lower concentration of substrate. When the amount of enzyme is reduced, one must have more substrate to supply the reduced amount of enzyme sufficiently to get Vmax by 2. The next one. Enzymes involved in the electron transport system are mainly located on the inner mitochondrial membrane. tDNA carries which of the two phytohormone biosynthetic genes? So it is auxin and cytokinin. Then average length of CC single bond is 0.154 nanometers. Horizontal gene transfer in bacteria can occur through. So horizontal gene transfer is the movement of genetic information between organisms. So it can occur through conjugation, translection and transformation. Then next one. The main role of carbohydrate in the cell membrane is. So carbohydrates are major components of the cell membrane placed on the outermost surface of the cells bound to lipids or proteins. The membrane carbohydrates are involved in cell adhesion, recognition and act as physical barrier. So molecular recognition is the answer. Then shrinkage of protoplast from cell wall due to exosmosis caused by hypertonic solution is. So it is plasmolysis. That is, it is hypertonic solution. The cell loses water and become plasmolyzed. The next one. Which of the following antibiotics act by inhibiting bacterial cell wall synthesis? It is penicillin. It acts at the transpeptidation stage of cell wall synthesis to inhibit peptidoglycan cross-linking. Then choose the epimers from the following pairs. That is, glucose and galactose are epimers. Then in continuous fermenter reaction, the phenomenon of washout is obtained when dilution rate is more than maximum specific growth rate. Answer is option 1. Which of the following class of enzyme do not participate in the glycolytic pathway? Isomerases participate, lyases, transferases also participate. The one which doesn't participate is ligases. Option 2. Most mitochondrial empty DNA is inherited by the egg cell. It is maternal inheritance for mitochondria. Then, there is a mutation in the lac operon repressor in E. coli which prevent it from binding to the operator. So, naturally, beta-galactosidase will be synthesized because the repressor doesn't bind to the operator. Now, what is likely to happen when level of glucose is low in the cell? Now glucose levels are low means CMP levels are high. The CMP attaches to CAP allowing it to bind to DNA. This helps in binding of RNA polymerase to the promoter resulting in high level of transcription. So it will result in constitutive expression of lac operon genes. Then next one, CRISPR-Cas9 system can be used for editing nucleic acids. Then next one, the significance of a hit after a blast run is measured by E value. E value is expected value is a parameter that describes the number of hits one can expect to see by chance when searching a database of a particular size. Then base sequence variation between individuals that result from point mutation are called single nucleotide polymorphism. A point mutation occurs in a genome when a single base pair is added, deleted or changed. Then Baltimore classification of viruses is based on nature of the viral nucleic acid. Answer is option O. Then next one. A cDNA encoding a human protein of interest was cloned in a bacterial expression vector and introduced into bacterial cells for expression. But no expression of the human protein of interest was obtained. This could be because codon bias. What is codon bias? It is preference for one codon over other codons. Example, alanine. One codon out of 4 is always preferred by the bacterium. When the non-preferred codon is present, it results in non-protein, no protein synthesis. So the answer is option 1, that is of codon bias. Then next one, denaturation of a protein leads to loss of biological activity mainly due to loss of secondary and tertiary structure. Answer is option 3. Then Rushton disk turbine is widely used in stirred tank fermenters to mix the culture broths. The type of flow pattern generated by it is radial flow. Answer is option 2. The next one, ProCheck. ProCheck is a tool to assess stereochemical quality of protein structures. Then next one, the primary enzyme responsible for duplication of the chromosome in E. coli is DNA polymerase 3. Then next one, which of the following is an exception to the law of independent assortment? When genes are close together on a chromosome, the alleles on the same chromosome tend to be inherited as a unit more frequently than not. Such genes do not display independent assortment and are said to be linked genes. Answer is option 1, linked genes. Which enzyme is involved in the repair of DNA damage induced by UV light? That is, thymine dimers are formed as a result of UV light damage. So the enzyme that is involved in repair is photolyase. Crystal structure of a protein is available with the following resolutions in the database. Which one would you choose for homology modeling and structure-based drug design? It is of 1.2 angstrom because it is the smallest and because of that resolution will be more clear. So answer is 1.2 angstrom. A, B and Z DNA are respectively. They are right-handed. A is right-handed, B is right-handed, Z is left-handed. Next one. Which of the following is a method to study time-dependent behavior of biological molecule? So what is molecular docking? It is to predict the preferred orientation of the ligand to its target to form a stable complex. What is molecular? That is orientation of ligand with its receptor to form a stable complex that you study using molecular docking studies. Molecular modeling, it encompasses all methods, theoretical and computational, used to model or mimic the behavior of large biological molecules. And what is molecular designing? Is to develop new synthetic methodologies and to create new molecules with different degrees of complexity and with different functionality. And molecular dynamics, simulation of proteins, allow protein motion to be studied by following their conformational changes through time. So the answer here is molecular dynamic simulation. Low expression of a protein is observed in a given tissue. Which of the following techniques will be used to determine whether the defect is at the transcriptional level or translational level? So low expression of protein means you can go for Western blot, and whether it is the defect at the transcriptional level can be checked by using Northern blot. So answer is northern blot and western blot. Alpha helices in a protein usually do not contain certain amino acids which are also called helix breakers. Identify the correct pair of amino acid that is generally not found in alpha helices. So it is glycine and proline which are helix breakers. Which of the following is correct for double-stranded DNA? Purines equal pyrimidines, A plus C is equal to G plus T, C plus T is equal to A plus G. So all the options are correct. Next one. In the context of process control, a stable system is defined as one for which the output reference is bounded by all bounded inputs. Answer is option 3. Then PDB contains information about. So PDB, it contains information about the tertiary structure. The protein data bank, that is PDB, it is a database for the three-dimensional structural data of large biological molecules such as proteins and nucleic acids. Then next one, an E. coli cell culture 1 ml was diluted 10 raised to 6 fold and 200 microliter of this was used for plating. After 24-hour incubation, the colony forming units was found as 150. The CFU count of the original. So the dilution factor is 10 raised to 6. Volume plated after dilution is 200 microliter. So 200 divided by 1000, that is 0.2 ml. Now CFU per ml is equal to CFU into dilution factor by volume plated in ml. So 150 into 10 raised to 6 divided by 0.2. So that will be 7.5 into 10 raised to 8. Then next one. In most cases, immune cells that recognize antigen in complex with MHC they are cytotoxic T cells. Helper T cells recognize in association with MHC2. The next one. Polypeptide chains of the silk protein fibroin are predominantly in beta sheet conformation and has higher number of the following amino acid, that is glycine and alanine. Then next one. A fermentation broth with viscosity of 10 raised to minus 2 PAS and a density of 1000 kilograms per meter cube is agitated in a 2.7 meter cube baffled bank using Ruston turbine with diameter of 0.5 meter and stirrer speed of 1 per second. The impeller Reynolds number for the vessel is. So impeller Reynolds number, it is by definition the impeller diameter squared times the rotational speed time and the liquid density divided by fluid viscosity. So they have given fluid viscosity, density, rotational speed and diameter. Substitute in the formula and you will get 2.5 into 10 raised to 4. Then pyruvate dehydrogenase complex catalyzes the formation of acetyl-CoA from pyruvate. Which of the following describes the type of reaction that occurs? So it is oxidative decarboxylation. Then, in addition to tuberculosis, Robert Koch also known for the discovery of the causative organism for cholera. Next question. In microbiological studies, plaque assay is carried out for measuring the number of infectious virus units because it is used for mainly for quantification of virus particles. Then which hormone is involved in closing stomata? It is abscisic acid. An infectious RNA particle is known as a viroid. Which of the following technique is the most appropriate for resolving the proteome of a bacterial cell? It is 2D gel electrophoresis. In 2D gel electrophoresis, it separates the complex mixture of samples using two different properties of the protein. One is in the first dimension, the proteins are separated by pi value and in the second dimension by the relative molecular weight. Then which among the following is not an example of an RNA virus? It is SV40, it is a DNA virus. It is not an RNA virus. Agarose gel electrophoresis is routinely used for analysis of DNA preparations. Which of the following cannot be achieved by this electrophoresis? You cannot achieve DNA sequencing by using agarose gel electrophoresis. Then which of the following endonucleases is involved in post-transcriptional gene silencing? That is dicer. It plays a pivotal role in the initiation of RNA silencing, RNAi. by recognizing double-stranded RNAs and cleaving them into small RNAs using its RNase 3. The next one. To promote the attachment and spreading of anchorage-dependent animal cells, the surface of the culture vessel need to be coated with pronase. Answer is option. Then, in the presence of a competitive inhibitor, Km of an enzyme increases. Under aerobic conditions, what is the net yield of ATP molecule from beta oxidation of one molecule of palmitic acid? Answer is 129. I have explained it in the next. So palmitic acid, it is made of 16 carbons, so 8 acetyl CoA and 7 NADH and FADH2 will be produced. Now 7 NADH will produce 21 ATP and 7 FADH2 will produce 14 ATP molecules, so totally 35. And each acetyl CoA will yield 3 NADH, 2 FADH2, 1 ATP or GTP through Krebs cycle. So 3 NADH will yield 9, 1 FADH2 will yield 2, so totally 11. Then 1 GTP or ATP is produced, so that will be plus 1. That will be 12 ATP. So 8 acetyl CoA will yield 96 ATP. So 35 plus 96 will be 131. But 2 ATP molecule have been used in the activation of fatty acid in the first step. So 131 minus 2, that will be 129 ATPs are formed. Then next one. Which one of the following compound has the highest phosphoryl group transfer potential? So it is phosphoenol pyruvate. Then next one. Which of the following diseases are transmitted by mosquito bite? Malaria, dengue, yellow fever and filariasis. In the enzyme catalyzed reaction, competitive inhibition can be overcome by increasing the concentration of the substrate. Any given competitive inhibitor concentration can be overcome by increasing the substrate concentration. In such case, what will happen is the substrate level reduce the availability for an inhibitor to bind and thus outcompete the inhibitor in binding to the enzyme. So answer is option 3. The process in which microbes are used to reduce the toxic industrial waste or effluent is termed as bioremediation. Then next one. The process of weakening a pathogen so that it cannot cause the disease is technically called attenuation. Answer is option 2. Xeroderma pigmentosum is associated with defects in nucleotide excision repair. Disruption of nucleotide excision repair pathway by mutation can cause xeroderma pigmentosum, and this syndrome is predisposing affected individuals to the development of skin cancer. You successfully cloned and expressed a gene coding for antigenic protein from SARS-CoV-2 virus. But when you injected this recombinant viral protein in mice to generate functional neutralizing antibodies, you failed to get any neutralizing antibodies. What could be the most probable reason? The reason is the protein was folded incorrectly. If it was in correct folding state, then the epitopes would be on the surface and they would be easily recognized by the antibodies. So, since the protein was folded incorrectly, you could not get any neutralizing antibodies. The next one. Terminator technology in plant has been developed too. So, Terminator technology genetically engineers plant to produce sterile seeds at harvest. This will prevent farmers from replanting harvested seed and forces farmers to buy seeds each season instead. The companies will gain profit as a result of this. So the answer here is prevent unauthorized propagation of seed. Then baffles are introduced in a mechanically agitated bioreactor to promote agitation and mixing of the fluid. Then hypophosphatemia is inherited as X-linked dominant disorder. A normal male marries a heterozygous female affected by hypophosphatemia. What proportion of the offspring will manifest the disease? So this is the male, small h for recessive condition. So it is an X-linked dominant disorder. So the male is free of the disease, whereas the female, even if she is a carrier, she will be having the disease because it is caused by X-linked dominant disorder. then these are the gametes that are produced and these are the offspring. So 50% of the son and 50% of daughters will be the answer. Then structural alignment is measured by RMSD. Then in lac operon, promoter and operator refer to. So they refer to binding sites of RNA polymerase and repressor. The next one. The protein coat covering the nucleic acid in a virus is called. It is called as a capsid. Seed dormancy can be broken by weakening the seed coat. This process is called scarification. Answer is option 1. Which of the following is an antigen presenting cell? So B cell, macrophage, dendritic cell. All the given options are antigen presenting cell. They are called as professional antigen presenting cells. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.